A big hello to all my future toppers out there. Yes, you are someone who is going to top the list with the consistency that you are showing in the videos. I was so happy to see a lot of comments out there where you were answering the questions that I asked even in mid-session. First of all, I want to say big thank you, and please, if at all you feel that these videos that we are doing helps you well, tell it to your friends, subscribe to the channel, okay, and like the video, comment down whatever feedbacks that you're having or whatever questions or answers that you want to give. So over here, I have Gayatri Nayak and Deepika Manohar, who basically went through the session, answered a question that I asked in the mid-session. I was so happy. You know what? There is no big crowd for biology. Okay, there are very few people who come for biology, and usually they come during your PU2 midterm or basically towards the KCET examination. Then it will be flooded with 30,000 views, 22,000 views and stuff like that. But I am immensely happy to see that you guys are starting to preparing from this point onwards. You know how important this is and how you can outshine anyone out there with the best preparation out there. Kudos to you children, and thank you so much for putting those efforts and commenting down over here. It means the world to me as well. Okay, so it's not like I'm craving for comments and things like that, but you know, as a teacher who have taught in offline session, you get it, there is an interaction with child and the teacher which makes it all fun. And here we just meet online, and there's only one way to communicate, and that is through comments and everything, and I want you to actively participate through that. Okay, so thank you so much Gayatri Nayak and Deepika Manohar. You are absolutely correct with your answers. Chasmogamous flowers show 3 types of pollination, that is autogamy, geitonogamy and xenogamy. Okay, now going ahead with Putta Rajakarthik, he said option D is correct for the homework question that I gave. So there is one homework question that I give in every session towards the end, and he got it correct. Kudos to you. And even Nagaratna Vadar, he said JJ ma'am, option D is correct. First of all, I'm so happy to hear JJ ma'am from you, and yes, option D is absolutely correct. I have Sinchana also over here who said that answer is option B, aerial branches. I would say Sinchana, you're partially correct, but with the efforts that you have put down, again kudos to you, and I'll explain what is the actual answer for this question right now. Okay, but before that, let's see where are we standing via the progress sheet. So this is our progress card. Okay, so over here we have completed L1, L2. This session would be the last theory session where we are on complete endosperm, embryo formation, seed, apomixis and polyembryony. Exactly what in that? We'll talk about how is the endosperm formed, okay, how does embryo formation take place, what are the different stages, the dicot and the monocot embryo. This is a very boring diagram in your NCERT. I'll try to make it interesting, okay, and different types of seeds, types of fruit, and the most twisted topic out there in this chapter, that is apomixis and polyembryony. Most of the children find it to be very confusing. I'll make sure that you understand everything, and for those of you who are not able to understand because of the speed, do not be worried. Today this is a fresh video, this is not pre-recorded. I'll make sure that I'm talking a bit slow so that you guys can follow up. Now for those children who feel that I'll be slow, you can just speed up by the speed tab out there. Okay, works. And if you feel that I'm still fast, again you can go to the speed bar and then control it. My dear children, if you study this whole chapter with me, I can guarantee I'm giving you 11 marks in your PU2 examination and total of 3 marks in your KCET examination. And if you want the document, you can go and join our WhatsApp channel. Link is there in the description box. Now, do you have to trust us blindly? No. Here we have proof. We have brought in so many rankers out there, and we are pretty sure that you can be the next one. So 171 rank is not a small deal. I tell it again and again in every single video because I want you to have the affirmation that yes, even we can do it. Okay, and I'm telling you, you actually could be able to do it. Coming ahead with the homework question that I gave. So in Commelina, flowers are produced on. So first of all, what is Commelina? Commelina, Oxalis and Viola. These 3 plants are examples of them who have 2 types of flowers. One is the open flower, which can have the female and the male reproductive part, and suppose this is the soil level that we are having. They also have closed flowers in there, inside which again you have the female and the male reproductive parts. So this type of beautiful open flowers that you see, you call them chasmogamous flowers, and the closed ones which are there, you call them clistogamous flowers. How do you remember? Clisto has Cl, closed also has Cl. So that is why you can remember. So my dear children, if you see, this part of the plant is known as the aerial part. Would you call it aerial part, which is above the soil level? And over here you will be finding root or root like stuff which are known as rhizomes and things like that, my dear children. So over here in Commelina, flowers are produced on what? On the underground rhizomes or root? Yes ma'am, I can find clistogamous flowers there. On the aerial branches? Yes ma'am, I can find the chasmogamous flowers all there. So A and B, both options are correct. So leaves is not an option. In D option I have an option A and B both. So can I go with the D option directly? Yes. So which is why option D is correct. Okay, so all of you who said that A is correct, B is correct, you all are partially correct, but the actual answer becomes this thing. Okay, and it's not necessary that always it's over here, it can be towards this level as well. Okay, the chasmogamous flowers can be above the soil, but just right above the soil. I hope you got it. Cool. So I hope that we are good to go, and we are going to have a little flashback session because I want you to understand what I'm going to teach today. So those of you who probably didn't attend the last session might not be able to understand it, which is why a small recap session. Okay, so previously, my dear children, what was happening? The egg and the male gamete were about to meet. Where? In the embryo sac of the female plant. Now, basically, the male gametes are 2 in number. How many number? 2 in number. Okay, so the first male gamete came and fused with the egg cell out there. Okay, and they gave rise to the small little zygote up over here. Okay, remember zygote is a single cell structure. So both the cells come together, joins, okay, they have a single nucleus out there, and what do you have? You have zygote. Fine. Now one thing that you need to remember is that basically, suppose I say a plant which is there, it has cells which contain 40 chromosomes out there. It is made up of cells which has 40 chromosomes out there. So basically how it might be formed, there will be a female gamete containing 20 and there will be a male gamete containing 20. These 20 comes together. Basically it gives rise to a zygote which has 40 number of chromosomes. This single cell zygote undergoes cell division. All of the cells that is going to form will obviously have 40 each. So always remember, the gametes which fuse, they would have half the number of chromosome, and when the gametes come together, whatever they're going to form will be having both sets of chromosome. Now when you say half number of chromosome, you have a term for it, and that is haploid, and it is denoted by N. So X cell has haploid number of chromosome. The male gamete also has haploid number of chromosome. And what about the zygote? Zygote would be having 2N. N plus N, 2N. That means it would be having diploid number of chromosomes. Okay, now over here, I told there are 2 male gametes. So first male gamete fused with the X cell right over here. Okay, how and all I told you in the previous session. So if at all you did not remember that, please go watch it if you have not watched it yet. Now the second male gamete will go directly to another cell out over here. So this is a very big cell out over here, and I have a name for it. It is known as central cell. Now central cell is 2N. Why, ma'am? How can it be 2N? So basically what happens is that this central cell is very unique because it has 2 polar nuclei. It has 2 nucleus. One nucleus has N, another nucleus also has N number of chromosome. So N plus N is equals to what? 2N. So central cell has 2N number of chromosome and fuses with the male gamete which has haploid number of chromosome. So basically over here, a diploid cell is fusing with a haploid gamete. Okay, diploid plus haploid, that is 2N plus N is equals to what? 3N. What do you get? It is the endosperm that you get, which is 3N in nature. This is the primary endosperm cell that you are gonna get. Okay, ma'am, why is this happening? What is the need for all this thing? Basically, this little zygote that you see, which is single celled, it has to undergo cell division, normal cell division, mitosis you say. Okay, can you say, okay, one homework question out there. homework question 1, mid-session. What is the type of division that the gametes undergo? Can you put homework number 1 and then answer the question? It will be really easy for me to understand. Okay. Now, my dear children, so over here, the zygote which is there, it has to undergo mitotic division and increase its number to form something that is known as embryo. Now, for doing that, it needs nourishment. Who nourishes it? It is the endosperm that will be nourishing the zygote to form the embryo. So you tell me, what forms first, the embryo or the endosperm? What forms first? It is obviously the endosperm that will be forming first, which later on will nourish the zygote to form the embryo. So this is the small recap, okay? And now we are going to talk more about the endosperm first, and then we'll talk about the embryo. Okay, so what about the endosperm? Why does endosperm develop first? To provide what? Nutrition to the developing embryo, or for basically the transformation of zygote to the embryo. Now, the primary endosperm nucleus. What is primary endosperm nucleus? So, my dear children, over here, basically, when this cell and this cell came together, okay, you got the primary endosperm cell. But what about the nucleus? There are 3 nucleus coming together, they all join. Over here, you have the primary endosperm nucleus. There is one nucleus out there, that is what? Primary endosperm nucleus, which would be, okay, just a second, okay. So anyways, which would be containing 3n number of chromosomes. So this is PEC, primary endosperm cell, and this is PEN, primary endosperm nucleus, which is being formed over here. Now what happens to the PEN? That is a question. So it divides repeatedly, forms triploid endosperm tissue out there. So basically this particular cell which was there, okay, with 3n, it is now gonna divide. There will be multiple nucleuses out there. Each nucleus would be surrounded by cell wall. They would be having their own membrane out there. There will be some nucleus, okay, out there which is not surrounded by anything. So all of them have their own nucleus. Okay, so this is the nucleus that they are having, but there will be some nucleus which are not going to be surrounded by any cells out there. So you have 2 type of endosperm being formed. Okay, first is free nuclear endosperm. PEN, that is primary endosperm nucleus, undergoes nuclear division. The nucleus which was there, it is undergoing nuclear division, but the cell wall is not immediately formed around them. So this is what free nuclear endosperm. This thing is free nuclear endosperm. Later on, they will be probably surrounded by the cell wall and things like that. But as of now, there is no such thing. Now, cellularization, that is cell walls form making the endosperm cellular. So over here, you see the other cells which are surrounded. So they are a part of cellular endosperm. So free nuclear endosperm and cellular endosperm. Always remember this free nuclear endosperm. Later on, they will be surrounded by the cell wall. Where can you exactly see this? In coconut. So if you have seen coconut, if you have broken it ever, you would be finding there is water inside the coconut that you drink, right? The coconut water that is there. What is it actually? It is just the free nuclear endosperm. And the white thing that you eat, what is that? That is the cellular endosperm. Ma'am, why is it actually there? So basically, if you keep it like that, okay, if you keep the coconut without breaking it open and things like that, slowly you will be able to observe that there will be embryo formation within this particular space. In the empty space around there, there will be an embryo being formed. So endosperm forms first, then the zygote which is there, which is so small, it will develop into an, embryo out there. If you want, you can check out the picture. You can put down in Google, that is coconut embryo. So you will be able to see it. Okay, so always remember what initially happens. You had the PEN, primary endosperm nucleus. This primary endosperm nucleus, talking about the nucleus divides. Some of it are surrounded by the the cell wall and things like that. You will call them cellular endosperm. Okay, some of it are just freely lying over there. You call it free nuclear endosperm. Always remember the nucleuses that are here, all of them have 3n number of chromosome, because initially the nucleus that we had has 3n number of chromosome. So this they are undergoing the cell division. So obviously it will be what, 3n numbers. So they are all triploid, my dear children. Whatever you see over here, they are all going to be having 3 sets of chromosome, because we started off with 3 sets of chromosome. Remember, PEN was 3n in nature, like this. Okay, because it was this plus this. These nucleus and this nucleus that is there, all of them came together and we got 3n number of chromosomes out there in this particular nucleus. So first nuclear division happens, which you call as karyokinesis. What do you call it? Karyokinesis. Okay, once the nuclear division has taken place, cell wall formation would be taking place. Nuclear division and then cytokinesis and then basically cell wall formation out there. Okay, that is the usual thing that is happening. I hope that's clear and you will never forget that. Now, the fate of the endosperm. What will happen to this endosperm which is formed over here? It can be completely consumed before seed maturation. What do you mean by that? So basically, when the embryo formation takes place, when the zygote to embryo formation takes place, at this particular point of time, what happens is that this endosperm might be totally consumed by the growing embryo over there. Okay, by the growing embryo out there. And what does the embryo do out of? What does the embryo make out of the endosperm over here? So for now, you just think you are the endosperm. Okay, and I am the embryo. Okay, so basically while I am forming from you guys, okay, I am taking all the nutrition from you guys, I have become like this. Okay, I have become like this. I have my own storage packs over here. Okay, I am storing something over here. I have taken all things from you, but I am storing in my own way, in my way that I want to store it. And that way is known as cotyledon. That way is known as cotyledon, about which I am going to talk to you very, very soon. So there are few plants, okay, there are few seeds in which you can see that the endosperm is completely consumed to form 2 cotyledons out there, one over here and one over here. Okay, you will get a picture of it later on. Okay, now there are another set of plants in which they do not completely utilize the whole endosperm. So they make just one cotyledon and they would still have little amount of endosperm still persisting right there. So you have 2 scenarios, ma'am. One is complete consumed before seed maturation. Where do you see it? You see it in pea, you see it in groundnut, and you see it in beans. Ma'am, how do we remember? So basically the peas, if I try to break it open, I get 2 parts like this. The peanut, that if I try to open it, basically I get 2 parts like this. So whatever things are there which can be broken into 2 parts like this, basically what are these 2 parts out there? These 2 parts are none other than the cotyledons. They are storing them in the form of cotyledons, which are the 2 parts. Now if you break it open, you will see a little small thingy around over here. Whatever you open, what is that? That probably would be the zygote which is there. Okay, sorry, not the zygote, basically not the zygote, basically the future root and the future shoot out there. That is the thing. So it will be the embryonal axis which is there. You're going to study about all of it. Remember that this particular part can give rise to a whole new plant if you put it in water and things like that. Okay, and later provided with nourishment with soil and stuff. So basically they have their own cotyledons, which is this. So peas, groundnut, beans, which has 2 cotyledons, utilize all of the endosperm and they are known as dicots. That means 2 cotyledon. Dicotyledon as in 2 cotyledons are there. Okay, now in some, as I said, it persists in the mature seed. Okay, in some the endosperm still is there because they just have made 1 cotyledon out of it. Okay, and what are those? Castor, coconut and everything. Castor, coconut and also wheat, rice, my dear children, all of it which cannot be broken down into 2 pieces, my dear children, all of it, they are known as monocots. They are known as monocots and they have their endosperm still left out there. So castor, coconut, CC, and also wheat and rice that we eat. So all of it has some amount of endosperm still persisting. I hope at least a little clarity that you might have got about the endosperm. If at all you didn't get it yet, do not worry, I'm gonna make it more clear for you. So as of now, we had the endosperm. being formed. Why do you need the endosperm? For the formation of embryo. Now what does the embryo do? The mean embryo takes in all the nutrition from the endosperm and make cotyledons, either two or at least one of them, and some of the endosperm might be still persisting. That is the picture that you are having. Now let's go ahead all about the embryo. So my dear children, basically the location, I told you, you had your embryo sac. In the embryo sac you had the egg cell over here. Okay, what is this region where you have an opening over here? So basically you have two integuments. The ovule which is there, it has two integuments, the outer integument and the inner integument, and there is an open region through which the pollen tube comes and passes on the male gametes, right? So basically the fusion happens over here. The fusion happens over here of what? The male gamete and the female gamete out over here. So basically they are going to form the zygote over here itself. Where does the embryo formation take place? Over here itself. So can I say it is towards the micropylar region, this region where the opening is there, I call this as the micropylar region. Okay, so basically embryo formation happens at the micropylar region of the embryo sac. This is the embryo sac where all of this is happening. So for your clarity, remember always, okay, so this is the ovule that we had. It is there inside the ovary. It is there inside the ovary. Once this fertilization has taken place, I will not call it as an ovule, instead I will call it as a seed. What will I call? Seed. And the outer region which is there, the ovary which is there, I will call this as the fruit. Okay, cool. So this is now the seed where all of this thing is happening. Inside the seed you have the embryo sac where basically the embryo formation is taking place, and here you had the endosperm as well. Now adaptation. Zygote divides after endosperm formation to ensure nutrition. So zygote will start division only after making sure the endosperm has been formed. Now the zygote to embryo transition has certain stages. First thing, you have something that is known as proembryo. So this thing that you see is known as proembryo, where basically the zygote, the cell of the zygote has divided into two. You have what? Proembryo. And after proembryo, you have globular embryo where it looks like a globe out here. After globular embryo, you have heart-shaped embryo. My dear children, the cells are keeping on dividing and have different shapes, which is why we have different names. So proembryo, then globular embryo, then heart-shaped embryo, and then finally you would be having a mature embryo out there. Now do not be worried seeing the different names out there. It is not very important for you to know. Okay, at least if you understand the structure of a basic embryo, it will be fine, and I am going to teach you that. Okay, so first proembryo, then globular, then heart-shaped, and then mature. Is this important, ma'am? Yes, this comes as an MCQ question. You have to tell the number. Okay, in order you have to tell how does the formation take place. So there is a wrap. Okay, zygote pro, globe heart grow. Okay, I know it might not be that funny, but I am pretty sure it will help you when you need it. So zygote pro, globe heart grow. So grow as in it's maturing. So zygote is there, then you have proembryo, then you have globular embryo, then you have heart-shaped embryo, and then you have fully grown mature embryo out there. So zygote pro, globe heart grow. So this is the thing that you can remember if you want to always remember the sequence of how it develops. So my dear children, we are going to get ahead into the seed now, where what is forming? The embryo is forming, where what do you have? You have the endosperm and everything. Remember, initially in the ovule, what was there? There were cells surrounding over here. Those were the cells of nucellus. So for some of you who have not attended the earlier classes, you might not be able to relate. But I am telling you, inside the ovule, you had something that is known as nucellus. This nucellus cell are usually consumed when the embryo sac is being formed. The embryo sac is where you have the female gamete and everything up there. Okay, so this layer is usually consumed, but in some it might persist, and we are going to study about all of that over here. And initially the ovule had integuments, but when it becomes the seed, the integuments transform into some other layers as well. So there's a whole reconstruction that is going to happen up over here. Okay, so we studied about the endosperm, we studied about the embryo which is there. Okay, how the formation, how the thing happens, and now we will see this actual structure of the embryo out. So first thing that we have is a dicot embryo. Okay, So dicot embryo, my dear children, how does it look? I told you it takes all the nutrition and forms 2 cotyledons out of it. So basically, when you say an embryo, just like how we have a head and what you call leg region, similarly plants also have things. A region that will be giving rise to their stem. Okay, a region that will be giving rise to the root out there. Fine. So what do you call these regions as? You call these regions as, the upper region is known as the plumule. Okay, it is known as plumule. And the region that gives rise to root, it is known as the radicle. Okay, fine. Now, this axis which is there, which decides what is plumule, what is radicle and things like that, this whole axis that you see over here, it is known as the embryonal axis. Embryonal axis. This decides where do you have the plumule and where do you have the radicle, the future root and the future shoot. Now, where do they store all the nutrition out there? They have 2 big cotyledons out over here. So this is one cotyledon and this is the second cotyledon out over here. This is first cotyledon, this is the second cotyledon out over here. Now if we see this axis above which the plumule is being formed over here, okay, you call this as the epicotyl region. Okay, from here to down, from the point the cotyledons are joined to down region, it is known as the hypocotyl region. Hypocotyl region. So again I am telling you over here, my dear children, you had the embryonal axis, there is a region that gives rise to the plumule, there is a region that gives rise to the radicle, that is a future root. There is a point from where cotyledons arise. The point above the cotyledons attachment is known as epicotyl region, where you have the plumule. The point below is known as hypocotyl region, where you have the radicle. Got it? Now this is the picture that is given over here. Now one more very important thing, I am removing the root and the, did it go actually? No. Okay. I am just removing the root over here and the stem over here so that you don't remember it like that. Okay, so radicle out there, since the radicle has to go deep into the soil, my dear children, it is protected with something that is known as the root cap. Okay, now in your NCERT, they have a different picture, something like this. This is for you to understand. Okay, it looks like an angel or a butterfly or things like that. Now I'll make it something like this so that you can understand it better. So over here, my dear children, you had the you had the embryonal axis and over here, okay, you have both the cotyledons. So instead of one this side and one that side, they have made both of them together over here, and here they have not made an exact big region to make the plumule, but I have made for your understanding. Okay, it's the same cotyledon which was there like this, now they are sleeping. Okay, so this is the epicotyl region, this is the hypocotyl region. Over here you would have the plumule, over here you would have the radicle, and over here you would be having the root cap. What am I talking about? I am talking about dicotyledons. This is a dicot embryo. What is it? It is a dicot embryo. What do you mean by embryo? Ma'am, zygote was there, the embryo is there, the embryo will give rise to the whole plant out there. Now, basically when the plumule will grow, it will take nutrition from these cotyledons. Okay, it will take nutrition from these cotyledons out there, and then only it will be growing up. Got it? So embryonal axis with 2 cotyledons, epicotyl is the above region of the cotyledons which has the plumule, the stem tip, okay, or the future shoot out there. Hypocotyl below the cotyledons, radicle is there, the root tip or the future root out there, root tip is covered by the root cap. This is about the dicot embryo. Always remember, this picture is important, you might be asked to draw it as well for your PU2 examination. Now going ahead with the monocot embryo out there. So what exactly happens in the monocot embryo? Now again, this picture seems to be very difficult to understand, so I will try to make you understand in an easy way. Again we draw the same thing. We have the embryonal axis, Okay, now over here they just have one cotyledon. So this is that one cotyledon out there, and I told you they are not that mean people. They have little endosperm still left. Okay, so the endosperm is over here. You have the cotyledon over here. This is your cotyledon, and this region is the persisting endosperm out there. So then, since they have just one cotyledon, you know, when there is only one kid, they treat them so nicely. They have so things. So it also is being treated very nicely, and they have a very cute name. Okay, which you might not find very cute, but yeah, the cute name is scutellum. Scutellum is the name of what? The cotyledon. So there are people who name your pen, the pencil and everything. Similarly over here, this plant is naming its cotyledon scutellum. Okay, so always remember, it actually tried to make one more cotyledon, but it was not able to. It was not able to. So basically the degenerated cotyledon, which is not being formed up over here, okay, it's like a small one. I will call this as epiblast. I will call this as epiblast. This is also the other cotyledon, but it was not able to survive. You can think like that. Okay. Now over here, my dear children, ma'am, so basically you are saying that this is the epicotyl region and this will be hypocotyl region. Yes, you are correct. Ma'am, so this will be the plumule, right? That is the future shoot, and this will be the radicle, that is the future root. Yes, ma'am. Ma'am, what's there extra in monocot? They do not have one one cotyledon. They just have one extra endosperm. Apart from that, do they have anything extra? Yes, they have. What is that? Basically, to cover the plumule out there, they have a leaf primordia which is known as coleoptile. Okay, coleoptile. I know it might be difficult for you to remember all of the terms very soon, but I'm telling you, it is going to be easy if you listen to me carefully. So this is coleoptile, and how do you know it's on the plumule? So plumule is from the letter P. Here coleoptile also has the letter P. Okay. Now over here, the radicle which is there, ma'am, the root cap is there. Yes, ma'am, root cap is there. So that's enough, no? No. This particular monocot treats everything so nicely. It has given name to the cotyledon also. So do you expect it can just end up with the root cap? No. There will be one more layer down over here, and this is known as coleorhiza. It has two R, okay? It is known as coleorhiza, which is again going to cover the root cap over here. So it has R, the root also has R. Coleoptile has P, this also has P. You remember like that, it is going to be easy for you. Okay, so what is the extra thing? Over here, plumule is covered by coleoptile. Over here, root cap is again covered by coleorhiza. You have an extra endosperm. The persisting cotyledon that you are having, you call this as scutellum. Okay, you call that as scutellum. The one that was not able to grow, you call that as epiblast. It got blasted off. Okay, done. Now you see this picture. So I don't know why they made it so confusing, but yeah, anyways. So my dear children, this is the scutellum that you are having. The only cotyledon which is there, it is right over here. Okay, this is the shoot apex, as in you're talking about the plumule region, and it is covered by something that is known as coleoptile. Over here you had the scutellum. Similarly over here also you had one cotyledon which was not able to make it, that is epiblast. Now you go down, you will have radicle. The radicle is covered by root cap. The root cap is basically covered by coleorhiza. So over here one R is there, so probably my spelling is wrong. Okay, I'm so sorry, you can erase it. I remember it with 2 R, I don't know why. Okay, anyways. Oh, it had 2 R, I think that's a mistake over here. Not by me, but by the NCERT. Okay, anyways. So this is the thing that you have to remember. Will you be able to remember it? I hope you do. Do you think that I am able to make you understand things? Definitely let me know in the comment section. Okay, so monocot embryo parts. There is one cotyledon which is known as scutellum, lateral to the embryonal axis. This is lateral side, one side of the embryonal axis. Radicle and the root cap is enclosed in coleorhiza. Okay, epicotyl with the shoot apex and leaf primordia inside the coleoptile. Okay, shoot apex and the leaf primordia, as in the one that will be giving rise to the leaf. Both of them are inside what? The coleoptile region out there. Done. Okay, so this is about the monocot embryo. Okay, where is it all happening? Right over here inside the embryo sac, in the micropyle region, the embryo is being formed with the endosperm which was over here. Now, a lot of changes are happening up over here. We have to see the structure of a seed now. I told you the inner integument, outer integument, they will be changing up. There will be so many changes up there. So, let's now talk about the seed, its structure and its types. So, are we ready? I hope we are ready. So, the first thing, seed structure. Seed has seed coat. So, the integuments which are there, they are converted to seed coats. And what are the names? Testa and the tegmen. What do you have? Testa and the tegmen. And apart from that, what does the seed have now? Seed does not have the endosperm now. Okay, that is long gone. Endosperm basically helped in the formation of embryo. Now we have to talk about the embryo, proper embryo. So it has the embryo has the embryonal axis and embryo has the cotyledon as well. Okay, embryo has the embryonal axis and the cotyledon. So whatever was there before, you forget everything. The zygote, the endosperm, those were the previous things. Now it is being forming up. So seed has seed coat, testa and tegmen, okay, which are basically formed from the integuments which are there. Now inside the seed you have the embryo which has the embryonal axis and the cotyledon. Why do you need cotyledon? For food storage, and it is swollen in leguminous plants, you know, like beans and stuff like that, they have it swollen. Okay, they have big cotyledons out there. Embryonal axis, it includes, okay, that does not make any sense. It has the shoot tip and the root tip, that's all. Micropyle. Okay, so you remember I told you there is an opening from which the pollen tube comes in. This opening is still over there. So testa and tegmen are right over here. Okay, testa and tegmen are right over here, but still you have an opening here, and that opening is still known as micropyle itself. Well, this small pore is for oxygen and water entry during germination. What do you mean by germination? Basically, this embryo which is there, it has to give rise to the shoot and the root. That process is known as germination, and germination would be only happening when you have enough amount of oxygen and water. Now, when the water and oxygen enters, there are so many hormones that becomes active, and that's when the plumule will be growing, the root will be growing and things like that. So, remember that as well. Okay, now there are two types of seeds, non-albuminous and albuminous. Non-albuminous means the endosperm is completely used up. Same like dicots. Okay, I told you in dicots what happens, my dear children, all the endosperm is used up for making what? Cotyledons. So those plants, okay, so always remember, dicots are the ones which are non-albuminous. That means they do not have the endosperm still left back. Okay, now you have the monocots. Monocots have the endosperm left back, so they are known as albuminous. Now, can you tell me example of non-albuminous plants? Ma'am, whatever example you gave for the dicots, remember you spoke in the endosperm part? Okay, over here, you told about the endosperm that where it is completely consumed, they are dicots. So, pea, groundnut, beans which can be broken into two, all of them are non-albuminous. They do not have any endosperm left back over there. And the one you just have which cannot be broken into two, they are albuminous, such as what? Castor, coconut, beet and rice. So, over here whatever examples are there for dicot becomes the example for non-albuminous, and whatever examples are there for monocot becomes the example for albuminous. Okay, cool. Now, so I'll just read out just for your understanding. Non-albuminous, endosperm completely used up, for example pea, for example groundnut. Pea and groundnut you can break into two, right? Albuminous, endosperm partially remains. Okay, example wheat, maize, barley and castor. So apart from wheat and rice and castor and coconut, you have barley and maize also, which you can remember. Now perisperm is written. What is perisperm? So I told you, usually what happens, the nucellus which is there, it is being used up, but in some it still persists, my dear children. It will be still persisting, and this nucellus which is very huge, you know what, it will be helping in the nourishment of the embryo out there. Okay, it will be helping in the nourishment of the embryo out there. Now this persistent nucellus which is there, you call this as perisperm. Okay, what do you call this as? Perisperm. Fine, this is what the persistent nucellus out there. And where do you see it? You see it in B square. What is B square? Black pepper and beet. One very favorite question for KCET. Remember, all these questions are from your NCERT. These statements are taken from your NCERT. This would give you a lot of questions out there. So perisperm is what? The persisting nucellus which is there, which wants to nourish the embryo out over here. And where do you find it? B square. That is black pepper and beet. Cool. Now, when seeds mature, its water content is reduced and seeds becomes relatively dry. Okay, 10 to 15% moisture by mass. That is a general thing. The general metabolic activity of the embryo also slows down. Okay, so when the seed is going to be mature, it becomes relatively dry, and my dear children, what happens? The metabolic activity also slowed down. Later on, when water and everything comes in, that's when the germination and a lot of things would be happening up. Usually it is in a dehydrated state, as we see. Okay, we usually have the chana and all, we put it in water and then only it gives rise to things, right? So why is it so? Because it has lost all the moisture and stuff like that. It's waiting for right atmosphere to grow. So when we put it in water, it has got enough atmosphere, good atmosphere out there. That's when it gives rise to the plumule and the root out there. Okay, so my dear children, we transformed from zygote to embryo. We needed the endosperm to do so. In some, the endosperm was completely utilized. You call this non-albuminous seeds. Where do you find it? Digotes. In some, my dear children, the endosperm is still persisting. You call them albuminous seeds, and they are what? Monocots. Fine, cool, done. We are going ahead. So over here I have a picture of the perisperm. So my dear children, I told you embryo, endosperm, it's there, that is separate thing. But apart from that, the nucellus which is there, that is also going to help. This particular thing, persistent nucellus which is there, you call it perisperm, and you find it in B square, that is beet and the what do you call? Beetroot, sorry, beet and the black pepper. Okay. Now, next thing, the advantages of seeds. So we spoke about seed, the types and out there. Okay, you have non-albuminous, albuminous, and you spoke about perisperm and everything. Now you talk, what is the advantage of a seed? Why is this important? PU2. Okay, for PU2 you can get answer, you can get question what is the importance of seed, and you never expect that coming off. So independence from water. Pollination and fertilization do not rely on water. Okay, that is one advantage of a seed. Okay, it is what basically you want to make it independent of what it can survive. Okay, it can survive whenever you get right moisture, that's when it will be growing. Dispersal and colonization. When you have seed, my dear children, it will be helping to what spread around to different area. If it's not a very big thing, okay, it's not a very big thing. So even if it is attached to one organism's body, it can easily get dispersed. Now it acts as a food reserve. So suppose, my dear children, the plant, if it does not have enough amount of food reserve, obviously it will die. But seed is something that have enough amount of food reserve with the anatomical structure that it has. Now it is also there for protection. You are basically protecting what? The embryo in there, which can give rise to a whole new plant. So it's for protection. Now genetic variation. Sexual reproduction creates new traits. Basically seed is a result of what? Sexual reproduction. Seed is when male and female gamete comes together. That's when you get a seed. So don't you think it can give, it is the one that is responsible for giving out more amount of variation, genetic variation? Yes. So these points might not make sense completely, but it's there in your NCERT. I just want you to go through it once. Okay. So those are the things. Okay. Even this point does not make any sense out there. If at all I can find a better way to explain it, I will be telling you. But as of now, at least these 4 points, you can have it in your head. Okay. Now seed longevity. What do you mean by that? There are seeds that loses its viability in months. As in basically even after months, if you put it in water, it won't be able to grow because it has lost its viability completely. Viability as in the ability to germinate and things like that. So you know what? Basically there are 2 plants out there who have beaten viability. After 10,000 years also, they were still viable, they were still able to give rise to a whole new plant. What is that plant, my dear children? It is known as Lupinus arcticus. And where did you find it? In the Arctic tundra, where there is too much cold, my dear children. Okay, so that is the story about Lupinus arcticus. And apart from that, you have Phoenix dactylifera. Okay, you have Phoenix dactylifera. It was viable for 2000 years. Phoenix dactylifera is nothing but date palm. So you tell me what is what. Okay, you have 2 pictures over here. Guess who is who. this is A and this is B. So over here I am pretty sure, okay, you're using your brain cells out there, right? So these are the date palm. So date palm is basically the Phoenix dactylifera. For how many years? 2000 years, my dear children. It was still there, and it was actually excavated. It was actually found out from King Herod's palace. King Herod, there was a king whose name was Herod, okay. So King Herod's palace near the Dead Sea excavation, it was found that Phoenix dactylifera seed was there and it still grew, okay. And if you talk about Lupinus articus, it is way more different than this guy also, because it stayed viable for 10,000 years. You see how beautiful do they look, right? So this is Lupinus articus, that is found in the Arctic region. That is Phoenix dactylifera, which is date palm, which was found during King Herod's period. Always remember that, okay, very, very important. Cool. Again, this is something that gives question in NEET also, in KCET also, and in your PU2 MCQs as well. Fine. Now, with that, we are done about seeds also. I know some of you might not be able to gather everything, so I'll request you to do one thing. Pause the video. Okay, pause the video and just go through everything that we studied. Just move the cursor like this and go through everything that we studied, so you get an internal idea of what we are studying over here. Okay, so as of now, we were talking about the seed. Seed is what? The ovule after fertilization. Now we are going to talk about the fruit, and that is the ovary. What happens to the ovary after fertilization? So let's talk about that. Over here, ovary becomes a fruit. Okay, and ovary has its own wall. When you were saying ovary, it had its own wall. This is the ovule which has its own wall. Ovary also has its own wall. Now over here, when it becomes fruit, the fruit's wall is known as pericarp. What do you call it, children? You call this as pericarp. I'm just removing this picture. Okay, now pericarp which is there, I can divide into 3 segments. Pericarp is different from perisperm. Okay, perisperm is persistent nucellus. So pericarp has 3 parts. First one is exocarp. Second one is mesocarp. And the third one is endocarp. Now if I teach you like this, will you remember? Never. So what do? We go over here. Oh my god, just a second. We go over here and we make a picture of my favorite fruit out there. So everyone, do you think you also have this as your favorite fruit? This is mango, for those of you who are not able to understand my diagram. Okay, so my dear children, this is the seed of the mango. Okay, and now, so this part that you see is the ovary. Okay, the previous time it was ovary. Ovary's outermost layer, it is known as exocarp. So the yellow peel that we peel off, it is none other than the exocarp. Now inside that, we have another thing which is known as the endocarp. Is it? No, it is not known as the endocarp. People usually do this mistake. It is known as the mesocarp. Remember it. The fleshy thing that we eat, usually out over here, this is known as the mesocarp, the middle thing. M for meso, M for middle. And now, the seed which is there, you might have seen that there is a very tough structure around it. This tough thing which is there, it is known as the endocarp. Ma'am, so this endocarp is the part of the seed? No. The seed will have its own testa and tegmen and everything out there. This endocarp that you see, it is the part of the walls of the ovary itself. They all are walls of ovary. Together you call this as pericarp. The one that we remove, exocarp. The one that we eat, mesocarp. The one that we chew and throw, it is known as endocarp out there. Together they become the pericarp. So these are the walls of ovary. Inside that you have seed. Seed will have its own walls out there. Cool? Fine. You might have seen the seed of, if you have not, then you should be trying it today itself. So mango, you break it open and then you see the seed. Seed you can remove, peel off the walls out there. Those are the testa and the tegmen. Cool? Now I'm going ahead. If you want to take a picture of this, you can, because this is an additional slide. Okay. So where are we? We are here. So fruit, pericarp forms the, okay. My dear children, there are types of fruit. It can be fleshy or it can be dry. As in fleshy, as in for example mango. In mango you are able to eat the mesocarp, right? In some it is dry. They do not have that fleshy layer. It is dry. For example, you have mustard, you have groundnut. Okay, they do not have anything fleshy out there. All the walls seem to be just like one. That's all. Okay, so two type of fruits, fleshy fruits and the dry fruits. But that's not more important. What is more important is this. True fruits and false fruits. Ma'am, what is this? There is scamming in fruits also? Yes, there is scamming in fruits also. So basically, what do you mean by true fruit? So my dear children, basically, when the ovary which is there, that becomes the fruit, okay, it is known as true fruit. Example, you have mango and you have everything out there. In all of that case, ovary is the one that forms the fruit only, okay. So that's not a problem. So they are all true fruits. Ma'am, can you give an example of false fruit? Yes, I can. If you talk about apple, my dear children, if you talk about apple, what happens is that you might have seen a structure like this. Again, I have to draw something which is not there. Okay. You might have seen, even Picasso failed in front of me at times. So over here, that's too big. I'm so sorry. But I want you to have an understanding, that's why I am, okay. Female part, male part, and the one where they are resting, it is known as the thalamus, right? Here fertilization has taken place, the ovule became the seed. The ovary was supposed to form the fruit. It forms the fruit. It forms the fruit. But along with that, this thalamus also contributes to becoming the fruit. So thalamus also becomes a part of the fruit and it becomes fleshy. Where do you see it? You see it in apple. Okay, you see it in apple, and this is one very important example of what? False fruit. Why false fruit, ma'am? Because over here, it's not just the ovary which is taking participation in forming the fruit, but also the thalamus, the other parts of the flower. Sometimes it's not just the thalamus, there can be pedicel also which can be part. Pedicel, this is the pedicel. It can also be part of the fruit. You get it? So these type of fruits which are formed from different parts of the plant along with the ovary, such as thalamus and all those things, are known as false fruits out there. Example, you have 3 examples in your NCERT which you have to remember. There are different parts that forms it. One is apple, the other one is cashew, okay, and the other one is strawberry. So you can remember A, B, C. So A for apple, B for berry, strawberry, and C for cashew. These are the examples of false fruits out there. I hope this is also clear. Now over here, you have a picture for that. I will zoom this in and it becomes all the more clear for you. So my dear children, this was the seed. This is the seed. I am telling you, this whole part is not the ovary. Instead, only this part that you see, this is the ovary. This whole part that you see is actually the thalamus. So if you cut that open like this and see from the top, it looks something like this. Your seed and everything is there. Okay, your seed is there. Basically, the tough portion around the seed is the endocarp out there. Then there is mesocarp out there, and then there will be no separate epicarp in general, okay, and then you directly have the thalamus around over here. So basically, in short, this part that you see over here, it's nothing but the ovary plus the ovule. Rest of the part which is there, it is actually the thalamus. So next time when you are eating an apple, please remember you are actually having the other parts of the plant as well. Okay, will you be able to remember it? These are the type of fruits. Now there is one more type of fruits, after which you will say that I am not studying biology, because I also felt the same. Okay, so that is parthenocarpic fruit. Okay, what do you mean by parthenocarpic fruit? So it is like it is like the miracle that is happening in biology. So over here, just imagine a scenario, okay, where you have the ovule. In the ovule, the egg cell is waiting for the male gamete so that the fertilization happens. Only if the fertilization happens, this ovule will be turning into seed, the ovary becomes the fruit, right? But over here, without fertilization, so egg cell is still sad. So without fertilization, what is going to happen? This ovary which is there, it still forms the fruit. How? Basically, there are certain hormones which are there, which generally is responsible for the formation of fruit. These hormones somehow become active because of so chemical interactions out there, some changes out there, which is why it does not need fertilization. It does not need fertilization to form the fruit. Ma'am, does it actually happen? Yes, it happens. Where? In the case of banana. Banana. In the case of banana, you might have seen there are seedless varieties of banana. Yes. Okay, why seedless? Because fertilization has happened or not? No. If no fertilization, do you think this ovule can become the seed? No. So obviously there is fruit, but no seed inside this. And people have started commercially utilizing this particular property of the plant by giving hormones. They put plant hormones, okay, such that before the ripening period becomes what a fruit out there. That is why you can get seedless varieties of fruit in the market. All of them, they are known as parthenocarpic fruit. Okay, they are known as parthenocarpic fruit out there. Always remember that. Not parthenogenesis. Parthenogenesis is something that is different. Yeah, we see in honeybees. Okay, this is parthenocarpic fruit, where without fertilization, because of some hormones out there, the the ovary is transforming into a fruit. Here, they would not be having any seed. Where do you usually find it? In banana. But people are commercially utilizing this for their own purposes as well. Okay, so we humans are been so lazy now, we do not want even seed inside our mouth when we have a fruit. So we say we want seedless varieties of fruits out there. So that is how it happens in the case of parthenocarpic fruits. It develops without fertilization. Example, banana can be induced using growth hormones, which gives seedless fruits out there. Okay, done. So that is about parthenocarpic fruit. So we studied about seed, we studied about fruit, we have true fruit, we have false fruit, we have parthenocarpic fruits as well. Now, we are gonna go. Children, you're able to understand? If at all you have doubts, I make sure I reply to your comments in the comment section. Whether it be big paragraph or whatever is there, I definitely try to do my part. So if you have doubt, do not shy away. You can ask me. If you understood everything, give me, ma'am, kudos and things like that. I'll be so happy. Okay. Now all about apomixis and polyembryony. Now until now, before going ahead, okay, I'll explain you a bit of things here itself. Until now, we were seeing the condition where the ovary makes the fruit without fertilization. Agreed? So there was no seed. This is known as parthenocarpy. Now imagine the next scenario where basically this is the ovary. I'm just zooming in the ovary, okay? And here is the ovule. Inside the ovule, we expect what? The female gamete. We expect a female gamete to be haploid. Okay, this should be haploid. But somehow, the female gamete out over here, it is diploid in nature. This is what? The female gamete, and this is diploid in nature. Do you think it needs another male gamete to fertilize it? No. We assume there is no need for external fertilization over here. The female gamete itself is 2n. So this particular female gamete itself is a zygote, can be forming a zygote, can form an embryo out there. Okay. Well, over here, without fertilization, what is happening? Embryo formation is happening. That is basically ovule is making up a seed without fertilization. How does it happen? As I told you, because of some reasons, reasons in the plant itself, the female gamete out there is already diploid in nature. It does not need another male gamete out over here to form the zygote. So since it can only do the work, it does the work, it forms the embryo out there. Now this is the ovule that you are getting, which transform into seed without fertilization. This process of formation of seed without fertilization is known as apomixis. Why does this happen, ma'am? Because of so reasons. There might be some chemical coordination out there in the particular ovule out there. That is why it happens. Okay, unbalanced division out there. That is why it happens, but it happens. Okay, now one more term is given in your NCERT, and that is polyembryony. This is not separate thing. This is not at all a separate thing. I want you to understand that. It's same like this only. Okay, it's a part of apomixis. So apomixis is basically formation of seed without fertilization. So polyembryony, what happens in that is that basically this was the ovule. I'm going to make a bigger ovule over here. You have your nucellus still persisting. Imagine your nucleus cell are still persisting out there. And nucleus cell, my dear children, they were normal cells, no? So they were already diploid in nature. They were already 2n in nature. These nucleus cell are gonna enter inside the embryo sac. This nucleus cell are gonna enter inside the embryo sac, and once it enters, my dear children, it is going to give rise to the embryo. Once it enters the embryo sac, it is going to give rise to the embryo out there. How many? 1, 2, 3, so many. This is known as polyembryony, where the nucleus cell enters the embryo sac, forming multiple embryos out there. Is it a part of apomixis? Yes, because here also, anyway you are forming the embryo, okay, without fertilization, that is why it is a part of apomixis itself. Where do you see polyembryony? You see polyembryony in citrus fruits, such as you have orange, your lemon and things like that. In the citrus fruits, you see polyembryony, okay, which is a part of apomixis itself. So apomixis can happen in this way or it can happen in this way. The second way has a name, and that is polyembryony. Okay, and usually in general, if you see, apomixis is found in two family Asteraceae, and I'll show you one more. Okay, so over here you have Asteraceae and grasses where you see apomixis to happen. Okay, basically I want to tell you one more very important thing. Is it a type of sexual reproduction? Can you call it a sexual reproduction method, my dear children? No. Why? Because in sexual reproduction you need the male and the female gamete to fuse. So can I say that apomixis is actually a type of asexual reproduction? And in the case of plants, you do not say asexual reproduction, you say vegetative propagation, or basically you just say vegetative form of reproduction. Can you say that it is a form of vegetative reproduction in the case of plants? Yes. And whatever plant is good, the embryo which is there, whatever plant is going to be formed, don't you think it would be exactly like this particular plant over here, which has the female gamete? Yes. So whatever good feature it had, the same feature that the baby would be having, right? So can I say it would be producing clones? Will I be wrong if I say that? No. Since it is a type of asexual reproduction itself, okay, I can say that whatever it produces would be like clones itself. What's the good aspect of it? The good aspect of it for the farmers is that basically you do not need another plant to fertilize it and things like that. Here fertilization is happening. Here basically without fertilization only you are getting the embryo, and that embryo is looking exactly like the parent plant which had all the good features. Suppose the parent plant are disease resistant and they are good in all the aspect. It's very easy for the farmers to grow this type of plants, right? Okay, otherwise what do the farmers do? Basically they buy hybrid seeds. Okay, they buy hybrid seeds. Basically, one plant, two plant fuses, you get seed out of it, and these seeds are very costly, and they put it in the soil, and in the soil it grows up. But when it grows up, my dear children, the next round some other plant will come and fertilize, and now that hybrid thing that you had is lost, because again reproduction is happening, some other plant and this plant, you get it. So there is no purpose of having hybrid seeds. Okay, it will be very good if you can you can get apomictic seeds as well. You get it, instead of the hybrid seeds, because hybrid seeds, one thing, it is costly. Second thing out there, as I said, when they grow up, these plants can pollinate with other plants, and the hybrid factor which was there, it can be lost. Suppose the other plant is not very good in feature, so that now the next plant might give rise to might not be of good quality. So whatever money you threw, it's wasted now. So instead, why not use apomictic plants out there, which basically does not need fertilization only. Okay, the plant, whatever you're looking for, that plant is giving rise to more clones out of it. So that is the use of apomixis in the case of plant breeding. So I have taught you everything about apomixis and polyembryony. Once we read it, it becomes all the more clear. So apomixis is seed formation without fertilization. You find it in Asteraceae and grasses. It mimics sexual reproduction, but is actually a form of asexual reproduction. Why do you say it mimics sexual reproduction? Because at the end you get an embryo out there, you get the seed out there. Okay, that is why it is mimicking sexual reproduction, but it is actually asexual reproduction. Types, where the diploid egg cell forms without reduction division, develops into an embryo without fertilization. So this egg cell which was formed, it is not undergoing meiosis to form the egg cell. Okay, somehow it was formed and now it is diploid in nature, and this is going to form the embryo without fertilization. That is one method that I showed you. Okay, over here, remember the diploid cell which was there, it was forming the zygote and then the embryo. Now the next method that you are having, where is it? Okay, that is nuclear cells, the cells outside. The nuclear cells divide, enter the embryo sac and develop into embryos. You find in citrus fruits and also in mango. Citrus fruits and also in mango. This is known as polyembryony. Polyembryony basically means, the term actually means when there are more than one embryo per seed. Okay, when there are more than one embryo, if you talk about an orange, okay, one seed will have multiple embryos out there. You can never imagine that. I'm not talking about multiple seeds. Multiple seeds which is there, sorry, the seed which is there, one single seed will have multiple embryos within it also. This condition is known as polyembryony. When does polyembryony happen? When this thing happens. Nuclear cells divide and forms the embryo sac. Okay, now the genetic nature, apomictic embryos are clones. Yes, I told you, they just look like their parents. Importance in hybrid crops. Hybrids lose traits if grown from the normal seeds. If they are grown from normal seeds, they are going to lose their trait once, or from, as I told you, it can fertilize with the other thing. Now, apomixis prevents segregation, allowing farmers to reuse hybrid seeds yearly. So apomictic seeds, if you are using them, my dear children, it will prevent segregation, as in it won't have different features coming in. Okay, the same feature which was there, it will be keeping on going on. So this is a difficult term to it. As I said, you can remember that. Basically, hybrids are very costly. Second thing, hybrids basically can lose their traits, okay, if they fertilize with the other plants over there. But in the case of apomictic seeds, it won't. There is no segregation that you see. Okay, so apomixis is clear. How does it happen? Diploid egg cell or the nuclear cells, through that. Now when you have multiple embryos within the embryo sac, you call this condition as polyembryony. Okay, fine. Now with that we finish off everything. So this is the example where the nuclear cells are getting inside and forming the multiple embryos out there. Now we are gonna do the questions. So find the mismatch over here. First one, false fruit other than ovary, thalamus taking part in fruit formation is false fruit. Is it a mismatch? No, it is correct statement. Parthenocarpic fruits, fruits developed without fertilizers. Fertilizers are chemicals that we put. Why do we need to talk about them now? So yes, parthenocarpic fruits developed without fertilizers, wrong. Fruits developed without fertilization. So this is the mismatch. Okay. Now true fruit develops from the ovary, that is correct. Seed develops from the ovary. Okay, so basically I think that's a typo error over here. So this is not fertilizers, okay, this is fertilization. Okay, so this statement would be correct. I think this is something that is totally wrong. Seeds develop from the ovary? No, seeds develop from the ovule. This is a mismatch. So I might have not read it properly, which is why I might have put it over here. This is the answer. D is the answer. Okay, seeds developed from the ovary. That is wrong because seeds are developed from the ovule. So this is not fertilizer, this is fertilization. So this would have been correct. Okay, next. Persistent nucleus is found in, persistent nucleus which is there for nourishment is found in black pepper and beet, B square. So both A and B, can I say that? So your answer becomes D. Okay, next question. What is common between vegetative reproduction and apomixis? Both are applicable to only dicot plants. Both bypass the flowering phase. Both occur around the ear. Both produce progeny identical to the parent. Yes, because this is vegetative reproduction basically means asexual mode of reproduction only, right? So and apomixis is also like that. So both produce progeny identical to their parents. They produce clones. So D becomes your answer. Next question. In the embryos of a typical monocot, the structure enclosing the leaf primordia and the single cotyledon are respectively. Remember, where is the leaf primordia? You had the plumule, the leaf primordia, and at the top covering the plumule, what do you have? It has P in it, coleoptile. Okay, that is coleoptile. And the single cotyledon in the monocots, the cute name, scutellum. So coleoptile and scutellum becomes your answer. Your answer is B. Okay. Cool. And now you have the homework question. This is homework number 2. Rudimentary second cotyledon in monocot embryo seed is called what? Rudimentary as in it is not completely formed, it is halted. The formation is halted, stopped. So rudimentary second cotyledon in monocot embryo seed is called haustorium, epiblast, obturator. What is your answer for this question? So if you're someone who went through the session really carefully, you might be getting up the answer. Okay, so please comment down your answers out there, and just to clear out the confusion, since it's there in my head, I just want to talk about this. So I told you polyembryony is a part of apomixis. Remember that. Okay, why a part? Because over here when you have multiple embryos being formed like this, it usually happens in the case of apomixis only. Okay, it can happen otherwise, but mostly it is related to apomixis only. Polyembryony is a condition of having multiple embryos within the embryo sac. That's all. Okay, now with that clearing up, I would like to say thank you for being with me throughout this journey, and I hope that this time I taught you little slowly. If at all you still feel that, please, as I said, you can change the setting in the settings bar up there. Okay, and if at all you understood what I taught you, definitely let me know in the comment section. And if you are someone who is preparing for KCET, do not forget to subscribe to this channel, because we are doing our best and we have done our best in the past, and these are the results. Not just this, there are n number of people out there, and all of it, I challenge you, none of it you can find to be a paid person out there. So please understand that and do accordingly. And also I wanted to tell you one thing. Yeah, God bless you. Bye-bye. Take care.